First of all, I'd like to thank the 3D-SIG organizers for giving me the opportunity to talk our recent research of using deep convolution neural networks to improve protein contact prediction. Given a protein structure, we can calculate the distance between any two amino acids. If the distance is below a threshold, such as 8 angstrom, we say these two amino acids are in contact. And we can represent all the contacts in the protein using a contact map. For instance, residue I and J are in contact as a dot here, otherwise it's blank. And this contact map can nicely capture the structure patterns in the protein. For instance, here, this protein fold into first strand, then fold another strand. These two strands form a antiparallel beta sheet, and the contacts between them correspond to the contacts in these segments perpendicular to the diagonal. And they continue to form a alpha helices. The contacts in alpha helices correspond to a contacts in these, the strand correspond to the diagonal, and continue to fold in strand 3 and 4 correspond to the contacts between these contacts in this segment. And finally, strand 1 and 4 form long-range contacts, antiparallel contacts correspond to contacts in this area. And more interestingly, once we have sufficient number of good contacts, we can actually build a three-dimensional structure from these contacts. So then the key question is, contact prediction is, given a protein sequence, can we predict if any two amino acids I and J are in contact? That means their distance is below a threshold such as 8 angstrom. And we are only interested in the non-local contacts. That means the sequence separation between I and J is greater or equal to 6. And contact prediction has long been conceived. In 1994, people used correlative mutation between two amino acids in a protein family to calculate contacts. And since then, from 1994 to 2010, a lot of machine learning methods, statistical methods have been developed to improve contact prediction. However, they didn't receive a lot of attention because the accuracy is not high enough to build a three-dimensional structure. The first major advance came in 2011. People use co-evolution between amino acids in a protein family to predict contacts. If you have sufficient number of contacts, you can sequences, you can calculate the co-evolution, you can make a very accurate contact prediction. Another major advance is 2012 in CASP-10 experiment. Deep learning was introduced to improve contact prediction. And since then, in CASP-11, 2014, CASP-12, 2016, contacts have been shown to significantly improve tertiary structure prediction. And it actually is one of the most important major breakthroughs in the field of ab initio structure prediction in the last 20 years since fragment assembly was designed in 1997. Now, let me briefly introduce the two major advances. The first is direct co-evolution coupling analysis. If the two amino acids are in contact, in a protein family, if one amino acid is mutated, another one has to be changed accordingly in order to maintain the contact. So we can give a family of protein sequence, multiple sequence alignment. We can calculate the direct co-evolution between two amino acids and use this information to predict the contact. And the key is calculate the direct co-evolution, not the indirect one, not the accidental one. So that's a major breakthrough. And later, people use this information with neural networks to improve contact prediction. Another major breakthrough is technology. So in 2012, deep learning was introduced to predict the contacts. Basically, given a protein sequence, I can take one window of information around the amino acid I, another window around the amino acid J. I use them as input for deep learning network to predict if they are in contact. However, this method still treats each contact independently, each pair of amino acid independently. But we know that contacts actually highly interact with each other. They're highly correlated. For instance, contacts in these parallel beta sheet are related to each other. So a deep convolution neural network was introduced to take into account the interactions between all the contacts to improve contact prediction in CASP-12 and later published in 2017. Now, follow these two major advances, I'm going to describe our two-level deep convolution neural network to improve contact prediction. Now, I first try to use an image classification to describe the idea of convolution. So, for instance, give an image. I only classify these images into different categories: dog, cat, houses, and so on. In order to do the prediction, I use a neuron called a filter to take a square of input of the image to generate a feature. I then move to the next square to generate another feature. So by doing so, I can generate the two-dimensional features. Basically, I try to looking for some geometrical objects like a circle, like a triangle, and so on. I can use multiple filters to generate multiple feature maps. And then these feature maps can be used as the input of next convolution layer to generate another feature maps. This is very important. I can continue to do so, generate more and more feature maps. So features will be automatically attracted from your original 2D input without human crafting. That's the key idea. Another idea is features become more and more global as you move along the convolution. For instance, feature here corresponds to a square window of the features in here. Each feature in this window corresponds to a square in the original image. So this feature going to correspond to a much larger area in the original image than this feature in the previous convolution layer. So the features become higher and higher level until I get some sufficient high level features, I use them to do classification. So that's a key idea of convolution. Revolutionize machine learning and image classification, voice recognition, everything. So now I choose you one video to demonstrate this. You see a filter move along to generate features by multiply the input with the weights. So generate 2D-dimensional 3D feature maps. Using this technology, we design a deep, fully convolution neural network to predict context map. So all the input information between any two amino acids are stored in a matrix, two-dimensional matrix of L by L. L is the sequence length. For instance, we use one matrix to store the co-evolution between any two amino acids in the matrix. We also store the secondary structure of amino acid I and J in the matrix, so as mutual information between I and J in this multiple sequence alignment, and even contact potentials can be stored. Each matrix is just like a channel of an image. All these are used as input to generate the 3D-dimensional features using convolution. And we use 16 filters to generate 16 feature maps. I continue to do this convolution 6 times, 6 layers of convolutions. And in the last layer, I only have one filter to take the last layer of feature maps as input, take one square input around amino acid I and J to predict the probability if I and J are in contact. So I get a fully probability contact map prediction. And each prediction here depending on the whole input matrix, not just the information around I and J, the whole matrix. That's the key reason of the success of the convolution neural network here. And I can use the structure I just described to predict the contact at any threshold. And in the first level of our network, actually we predict the user input to predict the contact probability at a multiple sequence threshold, 6 angstrom, 7.5, 8, 8.5, and 10. The reason is the distance between amino acids is flexible. So we want to account for this kind of the variation. Then these contact probabilities are used as input together with original input for the next layer of convolution network that I just described to predict my final contact map at a distance threshold of 8 angstrom. That's a standard in the field. And we train this 2-level network on 1,400 proteins with known contact maps. We validated on 196 proteins, and then tested on the blind test them on 3 CASP datasets. And we train on the GPU. Now, after we train this network, first thing we do is we check whether or not 2-level network will actually help. If I only use 1-level network with 8 angstrom threshold, I get accuracy about 69% for L divided by 5 long-range contact predictions. If I use 2-level network, but only 8 angstrom threshold in the first level, I get accuracy close to 72%. But if I use 2-level network, I get accuracy above 72%. So that means this architecture actually improves accuracy. The second thing we did is we try to see the impact of different features on the L divided by 2 long-range contacts. The first is if you use all the features, the accuracy, let's say, is about 6.2%. Then we remove each feature separately to see how the accuracy is going to decreasing. So you see, you remove different features, accuracy decrease, secondary structure, and so on. But the features that make the most loss of the accuracy actually is the co-evolutionary features calculated from the multiple sequence alignment. If I remove all the co-evolution features, accuracy actually drops from 60% to 38%. That means this feature is crucially important for this convolution network prediction. After we validated these methods, we blind test our methods on the CASP3, CASP datasets, CASP 10, 11, 12, compare with the best methods of the time in the CASP experiment, from DINCON1 to Raptor X. The accuracy is here. And also we compare our methods DINCON2 with the standard methods using co-evolution features with standard neural networks. You can see the accuracy actually is better, substantially better than all those methods on these 3 datasets. And after the publication, I noticed that Raptor X also used convolution network, has improved the accuracy to 56.8%, and our methods will also continue to improve now to similar level. Then we can demonstrate that deep convolution network actually reached the state-of-the-art performance. Now, after this experiment, now I'm going to show you one example to demonstrate the power of contact prediction. So here is the CASP12 target. Length is about 104 amino acids. Blue represents the true contact map, okay? And red represents the top L divided by 10, about 10 predicted contacts. You see, they overlap pretty well. That means actually the accuracy is 100%. I actually use this virtual link, black link, to represent the predicted contacts. You see the contacts here. Now, the contacts in this segment correspond to the contacts between this blue strand and green strand. Contacts here correspond to the contacts between these 2 strands. And the contacts here correspond to contacts between this green and the blue strands. And the contacts here correspond to long-range contacts between blue and red strands here. So that's top L divided by 10 predictions. If I increase the number of predictions, top L divided by 5, 20 predictions, you see I see more contacts between those strands. And I also see some new contacts between new strands, this blue and light blue. Accuracy is still very high. Then I select top 50 predicted contacts. You see I see more contacts between those strands, and accuracy is still pretty high for this example. Finally, I use these predicted contacts as input for a tool called Confold to build the 3-dimensional structure of this protein. I superimpose this 3-dimensional structure, the red one, with the predicted structure, the blue one. So you can see the second structure superimposed pretty well. And actually, MSD of the core region actually is 2.3 angstrom. The TM score is 0.67. That means the topology of this protein is correctly predicted just from scratch, from context, without using any templates from scope, whatever, PDB. And the reason we did pretty well on this example is 2 reasons. The first is deep convolution network. It's you can integrate all the information to make an accurate prediction. Secondly is actually co-evolution feature is very powerful because we have sufficient number of the homologous sequences to calculate that co-evolution. With that, I'd like to summarize my talk with a few observations. I think the deep convolution network can integrate multiple sources of information easily to make the contact prediction. And the deep convolution network can capture the dependence between all the pairs, all the contacts to make a contact prediction, and can take into account the protein of any length. We use the same architecture for protein of any length to make a prediction. And the deep convolution neural network can extract the global features and patterns automatically from your raw input information. And deep learning field is very, very highly active. A lot of new technologies can be applied to further improve contact prediction. And we also release the source code of our method at GitHub, and you are very welcome to check them out. Finally, I'd like to thank my students who did the work, Badri, Jesse, Jay, and Tianqi, and also the funding agency, National Science Foundation, NIH, for supporting this research, and thank you very much for your attention.
Any questions? Do you use either C alphas, C betas, or the side chains as the contacts in the training data? I would have thought that in contact prediction that using the side chain context, which is where the sequence is, would matter more than, say, backbone to backbone, where the side chains are facing away from each other, and maybe that's not a useful context.
That's a great point. Actually, we use the distance between kappa alpha.
Yeah.
So it's the center of the side chain to decide whether or not it's a contact or not.
Right.
Instead of kappa alpha, yes.
Sorry, say it again?
We use the distance between carbon beta atoms
Betas, okay.
rather than carbon alpha beta atoms.
So at least you get side chains kind of pointing at each other as opposed to...
Exactly.
Yeah.
And that's a key thing.
Have you compared them to see how much better you do with and without?
About 10 years ago, we used carbon alpha.
Yeah.
Because it didn't work well. Now we use carbon beta. Yes, that's a key reason. Yes, yes. Thank you.
So thank you very much for that. So in some ways, what you're seeing is essentially parallel what's happened in other industries with 2D convolutional neural nets and the improvements in facial recognition from images and so on. People are now moving on. So the question is, what do you think about 3D convolutional neural nets?
Wow. That's great. That's great.
Because that, I saw an amazing talk from NVIDIA, who makes the GPU cards for video games, and they are now doing 3D machine learning on convolutional neural nets and getting amazing results. And it's a question of how we represent protein structure within a 3D context to actually make the most of these kinds of developments. But it seems like there could be another parallel going on there.
Yeah, that's a great question, great insight. And actually, I'd like to share with you a secret of our recent research we are doing. We're applying 3D convolution network to classify proteins 3D structure directly to different categories. We actually use this to predict the mutation size, DNA binding size, taking 3D convolution, take a 3D structure as input. And actually, that project was recently funded by National Science Foundation. So I think that's one reason people get impressed.
I was asking because we're doing the same thing, so let's talk after.
Yeah, we haven't finished the research yet, but just share you with what we are doing, what we recently funded, just funded actually July 1st officially started. So I think your insights, that's crucial to moving into that direction, yes. Yes?
How user-friendly or how easy to use is the tool compared to the others that
Well, that's a great question again. So let's say, 5 years ago, very few people like to use a neural network. Actually, they hate neural networks because it's so hard to write neural networks. But nowadays, there's a package called the Keras, TensorFlow, Siena. Make it so easy for you to build a deep convolution network, all kinds of networks you would like to have. And even, people without computer science background can build deep networks and train them on GPU. So that's one reason is everybody is talking about deep learning, because it's so easy to use. In the old days, only accessible to the experts in the field.
But specifically, are you sharing the network itself or sharing the package that you develop?
We share the whole source code, of the package.
Including the weights?
Yeah, all the weights, even the programs used to train your network on the data, everything is open source. You can get everything. And all those packages are also open source too. Those are fundamental packages used to build networks are open source. So that's why this field is taking off. It's so exciting. Even planet scientists, people never use machine learning, talk about the deep learning. So it's an amazing thing in the field. Yeah.
All right. Do you have a question?
Yeah. Can you apply this methodology to model part of a structure where some of the structure is known and you could kind of fill some part of your contact map?
Yes, yes, yeah. Potentially, you can just use an input or contact map as input to predict high resolution contact map. Try to fill in some missing context. Yeah. I think that's certainly possible. Yes. Yeah. Yeah.
Last question.
Thank you. One quick question about the test of your method. If you use in your training set all but the topological class to which the protein that you want to test your method with, if you have done that, what's the result that you get? I mean, in order for you to achieve this level of accuracy, do you need to have in the training set the example, or you can actually predict something new?
That's great. That's a great question. Okay, so if your training data actually are similar to the testing data, you expect to get a high accuracy because they are similar. But that's not what our proposal for contact prediction. For contact prediction, we try to predict contacts with lower proteins. That's the hardest case. So we tested on the CASP datasets or the template-free modeling targets, ab initio targets, that have no basic sequence similarity with any proteins in your training data.
Sequence by structure similarity?
Even no structure similarity based on the CASP definition. So basically, some of them do not have any structure similarity with training data set, but some of them have some structure similarity, but it's impossible to recognize that as sequence similarity, structure similarity from sequence alone. So demonstrated this is a right direction for the ab initio protein structure prediction.
Okay.
Yeah.
All right, thank you very much. Thanks all the speakers of the morning. We'll have a lunch break and we reconvene at 2 p.m.